Hi everyone, my name is Dana.
I'm Richard.
I'm Natalie.
And we are Floc app. We are a team that is developing an app to count and size flocks at the agua clara plants.
So one of the first technical terms that we used was something called OpenCV, which is essentially a programming library which has a lot of functions which are really helpful for computer vision, which was really helpful in our project because essentially what we're trying to do is take images and count parts of them. The second term is Python, which is a programming language, which is very accessible, which is why we chose to use it. Third is the depth of field. This was important because the hardware that we used this semester was a camera, and after a certain point, the image starts looking really blurry, so you can't use it anymore. So one of the things we tried to do this semester was measure kind of the depth of field of the camera. Lastly is something called LabVIEW. This is essentially a platform for visual programming, and it was used heavily in previous semesters, but we tried to move away from it currently for a bunch of reasons that we'll explain later.
So to kind of go into why we are a part of the agua clara team, the main goal of Floc app is to create an app that will analyze data, which will eventually improve the efficiency of all the plants. At the end of the day, we should be able to take photos and eventually return how many flocks are in the photo and also the size of those flocks. And eventually we want to kind of get a relationship with that photo and how much coagulant needs to be added or how much should be lessened.
So one of the ways that we did this is essentially what we tried to do is take pictures of the flocks in real time. So due to fluid dynamics, small flocks don't really attach to larger flocks because there's no more room on the coagulant little blobs. So traditionally, turbidity sensors can't size the flocks. Essentially what they do is they use light, which doesn't tell us how large the flocks are. So it doesn't tell us how much coagulant we need to add or subtract. And the second issue with traditional turbidity measures is that it isn't automatic. It needs to be done manually, and it takes about half an hour. But with our app, it'll take only seconds, and it's much more efficient and uses less materials.
Yeah, so in the past, the Floc app subteam has developed two main things. The first component is a script that counts the in-focus flocks and identifies also the in-focus flocks. Now this involves a system of complex filters and thresholding. Thresholding is a procedure in which we can actually convert grayscale images to binary photos. So photos that contain only black and white pixels. Additionally, the second component that the team has built in the past is a GUI, or otherwise known as a graphical user interface. This was made last semester, fall of 2018, in the hopes of helping to make the application of scripts that we've written this semester and perhaps in the future semesters a lot easier for the Floc app development in the end.
So to kind of sum up more of our previous findings and what the team has done in past semesters, the team also attempted to build a nice interface with LabVIEW, and although it worked and performed a nice interface between a plant operator and kind of what we're trying to do, a few issues with LabVIEW is that the software is not free and it is very expensive, and we would not want to purchase that for all the agua clara plants. And it's also difficult to upload the data from LabVIEW to Post database. So kind of to change away from that, we've decided to use a Python script.
As an overview to our research process and method, we spent the beginning of the semester trying to familiarize ourselves with the previous findings. So that included reading over a thesis, which was written by a grad student last year or 2 years ago, and it also required us going over LabVIEW and a couple of other things that were done last semester. But the majority of our time was spent learning OpenCV. And finally, after that stage, we started moving into writing our own software, and then we started working on the hardware, which we'll talk about later.
So we got very familiar with OpenCV in order to take on this challenge. We learned how to import an image. We learned about the filters that OpenCV has and what it can do to change photos, such as binarizing a photo, and as Richard mentioned, doing a bit of thresholding, and morphing the photo in different ways. In addition, we learned about OpenCV's blob detection method, which was used heavily in past semesters, but we've moved away from it because it is not accurate, which we will explain later on.
So as Vina mentioned, previously semesters they used OpenCV to kind of count the flocks, but in OpenCV they relied heavily on this function called blob detector. But what it did is it only counted the very small and circular flocks, as you can see from the image, so it wasn't really effective for us because we wanted the entire flock.
So we made a few changes to the code to also deal with some of the issues. So because we wanted to use our own script that could iterate over all the pixels and count in size, we needed to make sure also that every flock was one flock and that there was no gaps in between, as you can see on the photo. So we changed up the dilations, meaning that we eventually filtered the photo more than one time to make it so that the flocks just, the broken up pieces morphed into one. So one issue with this is that we lost a bit of size, but we determined that size is relative and we can say which is a large flock and which is a small flock. So that is not an issue.
So we wrote our own script this semester to count and size flocks based off of images that we fed into the morphing and blob detecting scripts that were done in the past. Now here's some sample output that you receive if you run our counting and sizing script. There's four main components to the output, the first two being the most important. The first one simply tells you the number of flocks counted in the image, and the second one gives you an array of values that corresponds to each individual flock's size.
So going on to the second half of the semester, we had to deal with issues with our camera and troubleshooting. So originally, as you know, they used LabVIEW in previous semesters, but we were trying to move away from that. So we looked at Python solutions. Some of these included using OpenCV, but when we had issues with OpenCV, we also looked into other options, including company software and a few solutions that we found while researching how to access a camera through a USB port. We go much more in depth in later slides. So we did work with using company software, but one of the main things we did was also working with Python and OpenCV to access a camera. So as you can see in the photo, currently the video capture method is set to 0, meaning that we were able to access our camera's webcam, but we unfortunately could not access the camera that was inserted through the USB port. So after doing some research, we found out if we change that 0 to a 1 or -1, we can access different cameras, but unfortunately we were still having issues and we were not able to detect our camera. So we explored other options. Some things that we did researching including Pi AF cam and also video capture, but unfortunately both these options didn't work. So we started looking to maybe why our camera wasn't recognizing, or our computer wasn't recognizing our camera through hardware solutions.
Turns out the problem is simply that the most recent versions of the MacBooks don't have their Ethernet ports embedded within the USB ports that they have. And so we couldn't actually use the Ethernet to USB adapters that we were given to connect the Ethernet cable, the camera, to our MacBooks. So we decided to invest in a new adapter that takes the Ethernet camera and puts it and lets us put it into the Thunderbolt ports in the MacBook Pros that we have. And so in the future, we can actually connect the Ethernet cameras properly, and we can hopefully access the camera that way.
given now that we have this hardware solution, we should be able to use our script with OpenCV to actually access the camera, which will allow us to complete our future tasks.
Yeah, so in terms of future tasks, the first thing that we really want to focus on next semester is integrating our own data into the POST database. What they do is very similar in that they return data to the user to make the process more efficient, which is essentially what we're trying to do. So what we foresee happening is having like a little section of their database that will tell how large the flocks are, and accordingly how much more or how much less coagulant you should add or take away. And then the way that this should be done is through a timed Python script. So the script will take continuous pictures of the water to see how large or small the flocks are and make the adjustments accordingly.
So that is the end of our final presentation. If you have any questions or recommendations, please contact us.